Good morning, everyone. Welcome to the McCance Center for Brain Health Seminar Series. Today, we're hearing about how physical activity protects the brain cell by cell in Alzheimer's disease. On the screen, you have the SMS code to text for confirmation of your continuing education credit, as well as the required disclosure slides. And with that, I'm going to turn it over to Dr. Jean Bowman.
Thank you, Elizabeth. Welcome, everybody. My name is Gene Bowman. I am faculty in neurology and the clinical trials and brain nutrition laboratory director here at the McCance Center for Brain Health. And I moderate these McCance seminar series. And today we have the privilege of hosting Dr. Wrann, who is an associate professor in medicine at the Heart and Vascular Institute and here at the McCance Center for Brain Health at MGH in Harvard Medical School. She leads a research program in neuroprotection and exercise. She also has an affiliation with the Harvard Stem Cell Institute. She's a recipient of K99 ROO Award from the NINDS, the Claflin Distinguished Scholar Award, and ADDF Harrington Scholar Award. Her research is funded by the NIH, the Cure Alzheimer's Fund, and the Alzheimer's Association, and it's been focused on the beneficial effects of exercise and metabolism on the brain, and specifically secreted factors in exercise as potential drug targets. And of course, her work has been covered in popular news outlets such as the New York Times and CNN. Today, she's here to speak to us about a few of her most recent papers and how physical activity protects the brain cell by cell in Alzheimer's disease. Welcome, Dr. Wrann.
Thank you very much for the invitation and for the kind introduction. One housekeeping note, I still see people using this AI note taker. As far as I know, we are not allowed to use that at MGH, the Zoom function for that. So I would appreciate if whoever has it still on would turn it off so that we are compliant. Thank you very much. Okay, so and then I'll dive right in. People can see my screen, correct? Or do I have to share back? I forgot if we're still sharing or not sharing.
We see you.
Please share the slides.
You're not sharing yet. No.
Okay, all right, then we go back to that. My apologies, I thought because we had it in there. So.
Looks great.
Good, all right, so we're back on. So yeah, thank you very much. So I just have to do my disclosures. I hold patents on irisin, and I'm also a co-founder on the consulting to Avon Therapeutics. Yeah, and takeaway message first, exercise is good for your brain. And I hope in the talk today I can explain to you a little bit what's the data that we have in humans and in mice that is like these recommendations are based for, and also to understand, like to explain a little bit more what are actually the molecular mediators that we have identified so far. So there's a lot of data out there that exercise is good, and especially for improving cognitive decline in an aging population. This is one study I picked, which was actually performed at the McCance exercise at the McCance Center here. What I like about this study is that the participants were able to choose the exercise intervention. So for 12 weeks, they had to go up to this heart rate recommendations for 30 minutes a day. They came back after 12 minutes, and they had a nice improvement in their cardiorespiratory fitness, here shown as VO2 max improvement, and also their cognitive function improved mainly on the executive tasks. And they did, that was the team from Wrann and Westover did a nice job looking at the whole task battery. And something that I always like to point out is also there is a dose response. So the more people were able to improve their VO2 max, so the more fitter they got, the more improvement they also had in their cognitive function. This is not just in healthy aging population. We also know that increased or more daily physical activity staves off the risk to develop Alzheimer's disease. This is some nice data from the Rush Memory Aging Project, but there's many more data out there. So they showed, based on Fitbit tracking data, depending if you're in the top 10th or the bottom 10th percentile of daily physical activity, over the time course of 5 years, you were able to cut your dementia rate in half, which, thinking about what we have in our other clinical toolbox, this is a pretty large effect. And the recent meta-analysis looked at and said 4,000 steps is the minimum dose you need to see a reduction in dementia risk. And around 10,000 steps they saw, the maximum effect is 50%. So just to give people some guidelines, and again, also to again, thinking of patient populations that are sedentary, kind of giving maybe more attainable goals for them. If you look on an anatomical level, a lot of the effects that we see are actually happening in the hippocampus. You're all aware of the hippocampus as part of the brain that's important for learning and memory. And this was first shown by Kirk Egson and his group, where they did MRI imaging of a healthy aging population that either underwent an aerobic exercise intervention or a non-aerobic control. And over the course of 2 years, what you can see here is that you either grow your hippocampus by 2% or you shrunk it by 2%. So this is a non-insignificant number. And people always ask, is this all like new neurons that are bubbling up? Part of that is an increased blood flow, and then part of that is also maintained synaptic connection, maintained neurons in there. And, given that the hippocampus is involved in spatial learning and memory, they also saw in these an improvement in the spatial learning and memory task. This is a very new study. I think it just came out last week, actually also from an investigator at Mass General, where they were trying not just to use looking at, using imaging in addition to cognitive tasks to understand how physical exercise can either stave off the development of Alzheimer's pathology or not. And what they had shown is over a very long time study, I think was it 14 years from the Harvard Aging Brain Study, they showed that the baseline, so these were healthy, cognitive, unimpaired older individuals that they followed over these many years. So they showed that, as we already know, that cognitive decline, it has a significant interaction. So depending on if you're already have more amyloid beta in the beginning or not, you will cognitively decline faster. And they also had, which is pretty new, this part on the tau burden, where they, again, if you have low A beta, this is the two blue lines are basically the same. But if you already start out with high A beta, even though you're still cognitively normal, over time, you can you will grow your tau burden slower. So I think that's a nice study, given that they had these frequent imaging in there to give more, tie it more to biomarkers in Alzheimer's disease, the exercise effects. Then me as a basic mouse biologist, what is really good for us, we can model exercise in mouse models quite well. So this on the left is a study in aging mice, where mice, they were already almost 2 years old, which is like 70 years old, maybe even 80 years old in humans. They were exercised only for a couple of weeks before. And so these were not mice that were active the whole life. They got active or got a running wheel into their cage to do the exercise when they were older. And you can see this is a memory task. So the mice that are sedentary, I don't know why it doesn't say the sedentary in there, but these are the sedentary mice. So they need a very long time to find the hidden platform versus the one that got the exercise training. They have very much improved memory and they find the hidden platform quite rapidly. And then looking at Alzheimer's studies, different investigators have used different mouse models. This is from my colleague Si-Hun Choi here, where they used the 5XFAD mice, a model for familiar Alzheimer's disease. And they also showed that running exercise improved, in this case, a working memory task. Both of them are hippocampal dependent. There are many more studies I could show, but just to make the point. Something that we have tried in collaboration with Chris Wrann, who was at the Martino Center at the time. Now he's at the Cleveland Clinic. He is an expert in MRI imaging, and especially also this DTI technique, was to see, can we observe the same morphological or the same imaging changes in mice that we can measure in humans. So we had mice, they were healthy wild-type mice, they were exercised for 8 weeks, and then we did MRI imaging, either T2 weighted or the DTI imaging. And what we could show again is there is a significant increase in hippocampal volume, same what we observed in humans. And we also saw that there was an improvement in fractional anisotropy, so which is a surrogate for morphological changes for more ordered structure. So this could be more axon myelination, more synaptic structures. So which again shows that we can use imaging tools, same as human and mice, to kind of have more translatable studies to follow these two species. And we also then followed up with these mice in heart health, because heart health and brain health are closely linked. And we saw also that the microstructure in the heart has improved in the same exercise paradigm. Something that I thought very curious is when I started these kind of like, my journey into the brain and exercise is how important the running component is. So people before had reported that running is good, but they didn't really know why. And there were also reports that enriched environment is important. So an enriched environment is, as you can see, a cage with these different tools and there are little attractions the mice can interact with, like tunnels, things to climb on. And there were like two camps. One of them thought that maybe it's just that when your brain interacts more with the environment or there's more synaptic firing, more calcium, this is really what you need. And that's why it happens in the hippocampus. Whereas some other people were like, no, maybe the running is really special. So two of my colleagues, Henriette van Praag and Justin Rhodes, did similar experiments where they had the mice either in the standard cage, nothing in it, the running wheel, enriched environment with the running wheel, but they also had the enriched environment without the running wheel. And as soon as you take out the running wheel, here we're looking at newborn neurons in the hippocampus, so each dot is a new neuron. I should maybe mention this, running is a very strong stimulator to grow new neurons in your hippocampus. In mice, this is very well documented. In humans, we also have some data. It's a bit more difficult to measure, given that these usually post-mortem studies where we inject a molecular marker into these mice. But you can see like there's almost a five-time enhancement of the number of newborn neurons in this part of the hippocampus with the running. But as soon as you take out the running wheel, this effect is much, much weaker. So there's something important about running. So this could be some systemic factor released from the skeletal muscle, could be the cardiovascular system that's getting trained. So, and this really kind of underscored for me that there's like really a lot still to discover in the terms of the systemic effects of exercise. And here I just wanted to summarize a little bit on the cellular effects. What do we already know? Why is exercise is good for your brain? On the one hand, we have the de novo neurogenesis, so you can grow new neurons in the brain in certain regions. We do have synaptic plasticity and also de novo synaptogenesis, so synaptic connections that are already there are strengthened. New connecting connections are being made. Important when you think about a lot of the neurodegenerative diseases, neuroinflammation is reduced. We see an increase in cerebral blood flow, which all together leads, when if you use imaging technology, we see this increase in hippocampal volume, and that altogether leads to this improvement in cognitive function. So what we are specifically interested in is trying to find these underlying molecular mediators of the beneficial effects. And conversely, can we then harness these molecular mediators to treat cognitive decline in aging or neurodegenerative diseases? And this is some of our findings. So we were specifically interested in myokines, as I said, with the importance of factors released by the muscle. And other groups had already reported that these small peptide hormones that come out of the muscle and exercise can travel through the bloodstream, and they had positive effects on the fat, liver, bone, other organs, but also on the brain. And this is like including our work that showed this. The exercise hormone we are mainly interested in is irisin. It is cleaved from a precursor protein and released into circulation. So this is the small peptide hormone. This is the circulating irisin part. And we have shown that it gets increased in skeletal muscle and the hippocampus, and it circulates in higher levels in mouse and humans with exercise. And one of the studies that we did a couple of years ago, where we really tied it as a regulator of cognitive function, the irisin part. And this is kind of the summary. So what we showed is that using genetic knockout models for irisin, so we made a global knockout mice that doesn't have irisin anywhere in its body. And we first put it in a running exercise paradigm where we could show that wild type mice, so if you compare the black and the red line, they were able to greatly improve their learning in these Morris water maze. But if you look at the red and the blue, the green and the blue, those lines are almost overlapped. Then we also looked in aged mice, and you can see if you don't have irisin, so if you're a knockout for irisin, your cognitive decline is much faster, because at this age where wild type mice can still, perform in the novel object recognition task, the knockout mice cannot. And then lastly, we also crossed these mice to an APP Alzheimer mouse model, did a similar task there where, again, the knockout mice at this time couldn't perform the task correctly anymore, but the Alzheimer mice on wild type could. In addition, we showed that irisin was not just required in physiology for cognitive effects. But conversely, if you give irisin back as a therapeutic, you can improve cognitive function in AD mouse models. And we used an AAV to deliver irisin systemically. So we had an increase in plasma irisin levels. We did this at an age of 8 months where the mice already had gliosis, neuroinflammation, and some cognitive deficits. And after a 2 months treatment, we put them again in the Morris water maze, and we showed a significant increase in their cognitive performance in there. We repeated these experiments also in 5XFAD mice and saw the same phenotype. We furthermore showed that it looks like irisin itself can cross the blood-brain barrier, so it's a bit complicated slide. So we did show that irisin, if you look in the periphery, is obviously made in the liver where we put the virus in. We saw it in the plasma. But if you look in the brain, and these brains are from mice that it's the same what you do in industry for these blood-brain barrier kinetic studies, you perfuse these mice with a lot of PBS to kind of flush out all the blood, because we know the irisin is in the blood. And then you just measure the irisin in the tissue. And obviously it's still in the liver because this is where it's being made. But we also saw significant levels of irisin in the brain. And this data has been confirmed in other studies where they injected recombinant irisin protein and also saw the same thing. We don't know yet the exact mechanism, but this is very promising. Then we also looked at the which could be the possible irisin receptor. And there was a study from Bruce Spiegelman where I did my postdoc in, where they had published that irisin mediates effects on bone and fat cells via alpha V integrin receptors, or to be more specific, alpha V beta 5 integrin receptors. So we tested this in our culture model. So we cultured neuronal stem cells, differentiated them to become neurons and astrocytes. We always get both in these cultures. We use super resolution microscopy with an antibody again, where we incubated the cells with irisin that had a flag tag on it. And then we let the irisin bind, we fix the cells, and then we come back with antibodies. And since it's super resolution, so each of these dots represents one molecule of either an antibody against irisin or the alpha V beta 5 integrin receptors. And we did use an antibody against alpha V beta 3 as a control because it's a very similar receptor, but not alpha V beta 5. And what we saw, there was a significantly amount of binding of irisin on the alpha V beta 5 receptors on these cell cultures, but not the controls. And then when we looked which cells actually expressed the alpha V beta 5 receptor, we found it actually on astrocytes. This is what is shown here, but not on the neurons. Sorry, so the astrocytes are here in green and the neurons are in red. You see there's almost no pink on them. So this was kind of like in the first part, kind of like explain a little bit more how irisin is doing its job. This was followed by a very nice study from Rudy Tanzi and Suhun Choi's group here, where they used these 3D Alzheimer in a dish mouse model to even dig deeper into the mechanism, and again could confirm that it's astrocytes and the alpha V beta 5 integrin receptor that irisin is mediating its task. And what they saw, they also saw and they observed a reduction in plaque accumulation and a reduction in cell death. We have done some further studies kind of branching out from the amyloid plaque models to also look at tau models, and could show that using a mouse model for tau disease, it's the P300S model, is one of the most aggressive forms, that the same treatment, this peripheral treatment of the AAV vector, which leads to increased plasma levels, led to an increased performance in the SAB, the spontaneous alternation behavior, and also an improved performance in the Morris water maze and in the probe trial and the acquisition. And then lastly, Bruce Spiegelman and Ted Dawson had a nice study where we also collaborated on, where they looked in Parkinson's disease, and they induced Parkinson's disease or Parkinson's-like disease in mice by injecting preformed fibrils of alpha-synuclein into the striatum of the mice. And then the mice were again treated either with irisin or GFP as a control. And they performed either behavioral tasks or looked at stereology. And here what you can see, these are the dopaminergic neurons. So this is how the healthy substantia nigra looks like, when you treat the mice with the preformed fibre, you get a lot less. However, if you add the irisin, you actually preserve more of the dopaminergic neurons. They quantified in all different ways. And if you do motor tasks, you can see that they perform significantly better in 2 different motor tasks. So suggesting that irisin acts through a mechanism that is not just specific to, amyloid beta in Alzheimer's disease, but something that is more applicable to neurodegenerative diseases all over. Okay, so this kind of concludes the first half. Now I'm going to talk about this new study, which we just did, where we try to understand on a more systemic level which cells respond to exercise the most. Is there an exercise-induced transcriptional program? Is that similar or different in wild type and Alzheimer's mice? And also, how much will get rescued by exercise from this transcriptional phenotype in AD? And for that, we had mice, either wild type or Alzheimer's mice. We had them at 7 months old, which is a time where they start to get plaques, they start to get neuropathology, but we are not that late in the disease where everything is already full of plaques and neuroinflammation. And what we observe are just secondary effects. So we really wanted to come in when we can still modify part of the disease course and see primary players. These mice were again exercised with running wheel. They got BIDU injections so we can label newborn neurons. We performed cognitive tasking with them because we wanted to make sure whatever exercise we did was actually sufficient to improve cognitive outcomes. And then we dissected out the hippocampus and the dentate gyrus with the neurogenic stem cell niche in there, because we also wanted to be able to look at neurogenesis, which I had told you happens in this specific area in hippocampus, and performed single cell sequencing with the 10x mouse model. We then showed here in the Morris water maze that there was a significant improvement in both genotypes with exercise. We proceeded then to do the single cell sequencing. We got around a little bit more, like almost 110,000 nuclei. We got all the major neuronal cell types and the major non-neuronal cell types. I think it was a total of 11 cell types. We used known markers to group these cells. So this is kind of like, by now standard, but we were very happy because in addition to seeing just mature neurons, a lot of this were these mature granular cells, that's the main population. We also saw cells of the neurogenic lineage, mainly neuroblast 1, 2, and immature neurons. So one of the cell populations we were very interested in. We also performed histology, again, to confirm that there were morphological changes, and we can correlate our single cell transcriptional finding with morphological data. And what we could show is that we already know that Alzheimer's disease leads to an increase in microglia in hippocampus, but we could show that the exercise was sufficient to reduce the number of microglia. On the transcriptional level, we actually saw an increase with GFAP in there. When we counted the cells, it was not as obvious. And then also importantly, we did see an shrinkage in the average plaque size in these mice. Again, other groups had already shown this. For us, this was just important to make sure that if we do this extensive single cell analysis, we're actually analyzing the brains of mice that actually had really benefited from the exercise intervention. So, and this was the first thing that we kind of globally looked at. So we looked at the wild type mice. So WS is wild type sedentary, WR is wild type running, and we have the Alzheimer sedentary mice and the Alzheimer running mice. And we looked, how many genes were differentially expressed in these different cell types. This looked more or less similar between the groups. However, when we looked at the identity, so which are actually genes that are changed, you can see there was a significant program that was different in wild type mice than in Alzheimer mice, which was, I think, the first kind of bigger surprise from the data set. So we had a lot of things to start out with. So we decided to first focus on cells of the immature lineage, because we know that there's a lot of plasticity in this in the cell types. And so just to remind people, so when you want to make a new neuron, you start out with these stem cell-like cells. they undergo different changes. So they start growing, like making their outgrowths. They migrate through the cell layer, and in the end, they build a nice dendritic tree, and they connect to then majoring neurons. And there's a nice progression of known morphological markers with that. We were able in our single cell data set to reclassify this using known markers and also novel markers, and we were able to find basically, the neuroblast type 3 immature neurons and immature neurons. And when we confirmed this with staining, this is BIDU staining, where you inject the mice at the beginning of the running exercise, so you can see which cells had proliferated and survived there. We can see that in both groups, there was a significant increase with newborn neurons with exercise. Something that kind of stood out is when we did these more systemic analysis, which suggests again that this plasticity in the newborn neurons is really important, is when we did a driver gene analysis to kind of, where we were trying to understand in which cell types there's a lot of change and which are the genes that are driving the change. One of the cell populations that came up here in G were the immature neurons. So there was a lot of remodeling happening there. And again, some genes were the same in wild type mice with exercise, and a lot were different in between wild type and AD mice. When we did what is called a cell chat analysis, where you look how the cells are talking to each other. And what you do is you look at which cells express the ligand and which cells express the receptor, and then you can infer which cells are presumably talking to each other. And the more red you see, the more connections are being made with exercise in these cells. And you can see again that it's mainly the cells of the neurogenic niche which have these connections. And I should point out, these cells make about 15% of the total cells, but they had almost 90% of the connections going them to and from. So this is a lot. And then what we saw is that a lot of the cell connections that were downregulated in blue. So what happens is if you have a wild type mice and you compare it to a mouse that has AD, a lot of these connections are downregulated. And when you then exercise these mice, which is shown here, so you compare then the Alzheimer mice sedentary with the Alzheimer mice that received the exercise intervention, there's an increase in this number of connections. So again, showing on a systems level that exercise is able to restore some of the damage that's being caused by the Alzheimer. Next, what we did is we looked at all the cell type and all the different cell types. So it was cell type by cell type level, and asked what happens to the transcriptional program. So all in AD and in exercise. So all the genes that are showing here, like all the dots, these are genes that are significantly downregulated or not down, like significantly dysregulated in Alzheimer's disease. And then we asked from these genes, how much are they actually restored by exercise? So the more they are recovered, the more red they are. And then, if they have a thick border, they are significant. You can see that we found not in these two cell types, but we also had, very few cells in here. But you see in almost all the other cell types, we saw a large portion of genes that were restored by exercise. And now we have this nice catalog where we can look in every single cell type to kind of see what is the transcription program that can get rescued. And we hypothesize that some of these might be actually good drug targets. So we started out with also some validation tasks. This was something the reviewers wanted us to do because they said, who knows what all these lists of genes that you're getting. So for example, in the mature granular cells, what we did is for these genes, which we then called REC-DEG, for recovered differentially expressed genes. So sorry to reiterate the point. So these are genes dysregulated in Alzheimer's disease, but restored with exercise. We did find a lot of them when we looked in the published literature. We actually found that they were associated with AD, Alzheimer's disease, already previously, which is very encouraging, which means what we are measuring is probably relevant to AD pathology. So it's a good QC there. We performed then in situ. So we measured the RNA expression using a morphological technique with staining, kind of like similar to immunofluorescence. So each dot is molecules of RNA, and then we quantified this, and we could show, we did this for like 2 out of these REC-DGs, we can show there was a significant decrease in Alzheimer's disease and a rescue with exercise. Again, confirming that our data set is valid. And then, of course, I mean, it's nice that we can do all these things in the mice, but what does it actually mean for the human condition? And I have a great colleague, Dr. Bruno Benitez at the BIDMC, and he recently generated a very exciting data set. So he had 67 single-cell nuclei sequencing from 67 parietal cortex samples, males and females. And the interesting part in there is they didn't just have sporadic AD in there, they also had AD, so, or we also call familial AD, so they either had mutations in APP or the presenilin gene, which is exactly the same thing what our mouse model had. So we looked at his data set, and we looked at all his, we looked at all our Alzheimer genes and compared this, again, cell by cell type, which are also AD genes in his data set. And we found, so if you look at the bolded numbers here, we found that a lot of the AD genes that we identified in our data set were also AD genes in his human familial Alzheimer group, which again is very encouraging because again, it shows that this mouse data is relevant for the human condition, and also shows that some of these pathological changes that we measured in our mice in the hippocampus also occur in a different brain region in Alzheimer's disease, showing that there is translatability of these data. And again, we had this for all the different cell types. I will not show this. I mean, the data is published, so everyone who has their favorite cell type can go in there and see, you know, which of his favorite genes are showing up there. So when we now talk about some of the important players in Alzheimer's disease, for example, in microglia, one of the REC-DGs that we found is this cystatin C, which we found it actually in microglia and also in astrocytes. And it has been previously already shown that using mouse models that Alzheimer mutations that decrease cystatin levels occur in Alzheimer's disease. On the other hand, that increasing it, cystatin levels decreased AD loads in mice, and also I think believed in cultural levels. Again, confirming that the data set that we generated is very, you know, is useful. We also found, REC-DGs in the immature neurons. Again, this is a favorite cell type of mine because of this important neuroplasticity in there. And here we decided actually to go deeper and actually investigate some of these novel genes that hadn't been previously described with an Alzheimer component. So from our top REC-DGs, we decided to more investigate these 2 genes. And we first did this in culture. So we can make cultures out of embryonic, they're called neuronal stem cells or neuronal precursor cells. And then we can, you know, either have them in proliferating conditions, we can see that they proliferate, or we can also differentiate them into mature neurons and then see how healthy or, you know, how good of neurons we get in the end. And you can see here already knocking down either of these 2 genes leads to a significant decrease in proliferating cells. So the stem cells lose their stem cell likeness. And especially if you look here, you can see that even the morphology of the cells is greatly altered. They become like, you know, less stem cell-like and more like fibroblast-like. If you do differentiate these cells and we look with gene expression, you can see that they're actually lacking a lot of the important, you know, neuronal or synaptic markers. We even went one step further. We said, what happens if you then expose these differentiated neurons with, you know, synthetic A-beta 42 to kind of mimic Alzheimer's a little bit in a dish? Or what we also did is we collected conditioned media from neurons collected from Alzheimer mouse models, from an A-beta mouse model. So the conditioned media will have already, you know, A-beta in there. And this reduces the cell survival, and even in a greater extent in the cells that didn't have these important REC-DEGs. And then lastly, because the reviewers really wanted to know, oh, you used all these embryonic cultures, but your paper was on adult stem cells, it was in adult mice. Do you see the same thing in neuronal stem cells or precursor cells generated from the adult hippocampus? And a very long story, a lot of work later, we can say, yes, it's the same thing. We reduce the proliferation potential, we reduce cell viability, and we reduce the ability, when we differentiate them, to grow healthy neurons. And then again, taking it one step further, I showed you hopefully convincingly that this was really important for, in a culture model, but what about in vivo? So to do that, to this step, we used lentiviral injections into the dentate gyrus. So again, the dentate gyrus is where the neurogenesis is happening. We inject the lentivirus on unilateral on one side of the brain. I don't remember if it was, I think we switched between left and right to make sure there's no bias. We used stereotactic injections for that. We came back 5 weeks later. We can show that there was a significant decrease of the expression of the ATPIF1, which we wanted to knock down in the hemisphere that received the SH virus. But the expression of ATPIF was undisturbed in either the side that got the contralateral side that didn't get any virus, or the mice that received the control hairpin, which shouldn't do anything. And then importantly, if you then look at immature neurons here marked with DCX, there was a lot less. I mean, it was, reduced by, 60, 70%. So this is a pretty large effect, which again shows that, our data that we had, the, treasure trove that we generated has very interesting molecules that will actually, that are important for Alzheimer's disease, that are important for neuroplasticity and exercise. And then another cell type where we had looked at REC-DEGs and did more of these validation were the astrocytes. Here, again, we used astrocyte cultures, either treated with a cytokine cocktail to increase astrocyte reactivity to promote neuroinflammation in the astrocytes, or adding synthetic A-beta. And we could show here very nicely that knocking down these REC-DEG increased markers of reactivity in these astrocytes when you don't have these protective genes. So again, one more validation for important astrocyte biology. So we first looked at, genes, gene by gene level. What we now wanted to see, does something happen if you look at the cell types as a whole with exercise? So when we looked at microglia, we were able to, what we call then subclustering. So based on the transcriptional profile, you can kind of ask, are there cells in there that are more or less similar? And what came out is that we actually had a subgroup of microglia in our data set. And when we did genetic analysis, it became pretty clear these are these disease-associated microglia that had been previously described, which matches because we only saw them in the Alzheimer mice. However, what was the really interesting finding is that with exercise, it was not that we actually reduced the number of disease-associated microglia. What we did is we actually increased the transcription profile. So a lot of the markers that make their microglia and that are important for the phagocytic activity, the expression increased in the Alzheimer mice with exercise. And we confirmed this using also isolation of microglia from 5XFAD mice and qPCR. So again, the reviewers just wanted to make sure this is not an artifact. And I think that's very interesting. Again, if you think about like how maybe increased microglia phagocytic activity is beneficial in Alzheimer's disease with exercise, because we saw the decreased plaque size in these mice as well. So possibly one of the mechanisms. So when we then astrocytes, which again, I think it's a very fascinating cell type. So to just remind people, what do astrocytes do? So they're one of these important support cells. They're part of the blood-brain barrier. And we know that also in AD, they're dysregulated, but it was a little bit less clear what happens with exercise. When we used, again, our subclustering method, we saw that there was a subpopulation of astrocytes that actually were fewer in the AD mice. And there was a trend that actually when you exercise the mice, you restore the number of cells. when we did the genetic analysis, and you can ask which are the molecular markers that are most differentially expressed. So these are markers for these novel astrocyte types. We found these two markers, CMD1 and cadherin-4. As opposed to the microglia, where it was pretty clear this was a previously described cell type. Here, we looked through all the literature that was available, and we could confirm that these are not what people have called disease-associated astrocytes. They also didn't match with the profile of reactive astrocytes. And one reviewer was asking, hey, is it possible that these are like these radioglia stem cells because they kind of hide in the astrocyte cluster? And we looked at those markers. And again, it wasn't something that has been described before. So it's pretty clear that we have here a novel transcriptional state, which is quite exciting. But the field also has recognized it's not just enough to have novel markers, molecular. You also, like, what's about function? Like, does it actually do something for the cells? So that's what we then investigated in the next step. As I mentioned, cadherin-4 is one of the important markers. So we stained sections from these mice with GFAP to mark the astrocytes, with cadherin-4 to mark this cadherin high subpopulation versus the cadherin low population. And we also stained it with ICAM2 as a marker for the blood-brain barrier. We could confirm that there were high cadherin high astrocytes. So these are not neurons, these are bona fide astrocytes that have a high expression of cadherin-4, around 2%. I think our single cell data had it around 2 to 3%. So this was really good that we could confirm that. Next, we looked at the astrocytic domain. So how big the cells are, how trophic the astrocytes are. And on average, what happens in Alzheimer's disease, cells become less trophic, and the more the good astrocytes, to kind of, put them into categories, the good astrocytes tend to be more trophic with larger astrocytic domains. And that's exactly what we saw. The cadherin high cells had these bigger domains. And then talking about what could be a possible function of these cells, as I had mentioned before, astrocytes are a very important part of the blood-brain barrier, and these more trophic astrocytes are much or significantly closer located to the blood-brain barrier, suggesting, maybe some important function there. And we also did, obviously a gene ontology analysis, and again, we found a lot of, positive phenotypes in there, suggesting good function in there. Okay, next slide. Hello? Okay. What we then also saw is that if you look in exercise, this transcriptional profile was enhanced of these cells. So you can see here, these are the wild type mice. When in Alzheimer's disease, you lose these marker for these astrocytes. And then when you exercise the mice, it comes back. This is C is a bit different way to kind of plot those data to show that if we have actually this protective gene signature of these new astrocytes, that we, by the way, we decided to term them neurovascular astrocytes based on their location, that this is enhanced in these running mice. And then we validated again using a different transgenic mouse model, where we just isolate astrocytes with qPCR. And again, we could show that this transcriptional signature was significantly enhanced with exercise. And then lastly, coming back to the human relevance, do these cells also exist in the human? And the answer is yes. Bruno and colleagues also found it in the human data set. They found one subpopulation, the astrocytes, that was actually high expressing with cadherin-4, which again is very encouraging. And we're looking forward to further investigate the neuroprotective role of this novel astrocyte type. Oh, sorry. And lastly, what we also saw is there were less of these protective astrocytes in the AD, and in there's a protective allele that is closely regulated in there. We saw actually a reduction of the protective allele in the, hey, where are you going? We saw that there was more of the protective allele in this astrocyte cluster 1 than of the non-protective allele. Again, using what we have from human data so far, which is not that much, but again, everything points to a supportive role or protective role of this novel astrocyte. Okay, and then lastly, I just wanted to briefly mention the oligodendrocytes lineage that we also looked at. There has been data already before that, there was an interest in what happens to oligodendrocyte precursors and oligodendrocytes. And we could also show that similar to other studies that had looked to the plasticity, we also found REC-DEGs and plasticity in this cell type with AD and exercise. And the last gene I wanted to highlight is what we saw was, sorry, two points. My apologies, I thought it was our next slide. Similar to like neurogenesis, there's also oligodendrogenesis happening with exercise. So you have the OPCs, the oligoprecursor cells, when they become the pre-oligodendrocytes or committed oligodendrocyte precursors, and then they become the mature or the myelinated ones. And here again, we could find the subpopulation in our data set, and we could show that they actually were also decreased with Alzheimer's disease and increased with exercise. Yeah, and I think this is the last slide here. So this one marker gene that was quite interesting was the SCD2, which is the rate-limiting enzyme to produce important fatty acids for myelin generation, which is, we saw in the oligodendrocyte precursors, was a very strong REC-DEG. So it almost got back to wild-type levels with exercise. It was a trend in the mature oligodendrocytes, but it was a very strong there. So which is, I think, another interesting gene to look at. And other investigators already had, kind of seen that this was dysregulated in Alzheimer's disease, and increasing the levels of SCD2 was protective. Okay, so just to sum up this part of the presentation, so we were able to observe a cell type specific transcriptional response to exercise, which is distinct in wild type in Alzheimer mice. Exercise restored a proportion of these Alzheimer dysregulated genes with a unique signature, which varies by cell type. And these REC-DEG present potential therapeutic targets for AD, as we had shown with a lot of different orthogonal validations, just listed all here. And then lastly, I like to usually kind of come back to like, what does it all mean for me? When people then kind of ask, like, how do I know I'm, my brain's okay, or like what exercise is doing? And I always like to send them to the brain care score that was here developed at the McCance with the QR code, so that everyone can do a little brain check on themselves, or a lot of people are also asking for their parents or grandparents. So, things that you can do for yourself to improve your brain health. So the take-home message is, exercise improves cognitive function and prevents dementia. I showed you and hopefully convinced you that the exercise hormone irisin is a crucial molecular mediator of cognitive benefits in exercise and a potential drug target for AD or PD, and that our interesting single-cell RNA-seq analysis revealed novel mediators of the neuropeptide response of exercise. And I would like to thank the people on the team. There were a couple of very driven postdocs from my lab that have participated in the Nature Neuroscience study, Joanna De Rocha, and this were Sophia and Rashid. They were participating in the irisin story that I showed before. We always have a great team of research assistants and students in the lab that are doing great work. The single-cell study was done together with Nathan Tucker, who trained at MGH and is now at the MRI, who did a lot of the bioinformatics analysis, and other collaborators here. And I already had mentioned Bruno Benitez from the BIDMC is not here. And I'm always very grateful for the funding that make these studies possible. So, and I'm done, and I'm happy to take any questions.
Okay, thank you very much, Dr. Wrann, for that wonderful presentation on exercise effects at the cellular level and some human data. If anybody has questions, they can post them to the chat or raise their hand here. Yeah, Rudy, go ahead.
Christiane, beautiful talk, the whole thing from the introduction to the data, really wonderful. Thank you for that. And I guess I have a really general question that I've wondered a long time, and I think I've asked Sehun this as well. If you take irisin in the brain, what does
Yeah.
it contribute more to? Amyloid removal or neurogenesis? If you had to pick one, is it equal? Is it more one than the other?
I think so in the young mice, we saw a lot of contribution to neurogenesis, versus in the older and Alzheimer mice, there is not as much neurogenesis left anymore. So that's why I think the effects are less. What we see is we both see this neuroinflammation loop. And if you have young mice that don't have any amyloid pathology, obviously you don't see any effects on amyloid, but you see a lot of effects on neurogenesis, versus in the Alzheimer mice, there is not that much neurogenesis left, but you have amyloid pathology,
Right.
regulated there.
So follow-up question. So if you induce neurogenesis and BDNF and keep these newly born neurons happy and thriving,
Yeah.
do you think that despite the continued presence in a patient of inflammation, amyloid, tangles, everything else is still there, but you have new neurons and they're healthy and they're staying alive, should that help with cognitive decline or with cognitive performance?
Yes, I would think so. And I mean, the data from Sehun, I mean, that paper kind of showed it nicely, because they had both, right? They just made new neurons, and then they made new neurons in a healthy environment, which showed, if you have healthy neurons, that is helpful. And I think there are also other studies that have shown this. And there's more data now on humans coming out, where they look in single cell sequencing, where they also find reduction in neurogenesis, not just in aging, but also specifically if you have the same age group and you have Alzheimer on top, you have fewer newborn neurons.
Right.
There are studies that show that the remodeling that happens when you introduce new neurons has like what we call a network effect, right? It's not just the newborn neurons, but the neighbor network, the interneurons in there. So there are lasting kind of effects in the ripple wave that even can be there in Alzheimer's disease when you have fewer neurons. So I think it's still, given that we get more data that it's also valid in human, I think it's still a promising strategy compared to other ones as well. So, or still a viable strategy. So with all the data that I'm aware of.
Excellent. Thank you.
Okay, I see a question from Du. Du, would you like to pose your question in the chat? I see
yeah, I can tell.
Hi, welcome, Du.
Hi. So nice talk. So I'm wondering whether you were trying to dissect effects of irisin versus exercise alone. So there should be a irisin increased by exercise, and you do have all the data. Have you ever tried comparing back and forth? And second question is on the chat box, is that any study done by irisin knockout mice in Alzheimer without exercise? That's another direct proof that irisin does role in the exercise-induced Alzheimer reduce or not. And I wonder whether you see any of these studies.
Yes, trying to kind of disentangle to make it clear. So we did the irisin study, the exercise studies. We did this in the wild type mice. We did not do the exercise study on the Alzheimer mice, mainly just because the breeding is very complicated. They have very few litters, and it always takes forever to breed up, any significant cohort. So we haven't done what you're probably asking about. If you knock down irisin in an APP mouse and then you exercise, what happens? But we can already show that not having the irisin reduces cognitive function there. We have another neurodegenerative model. I can't say which one it is because it's not published yet, where we did show that, not having the irisin was detrimental. So if you exercise the wild type mice in this neurodegenerative model, you get huge neuroprotective effects. If you then exercise irisin knockout mice in this neurodegenerative context, there's almost zero effect of exercise. So in that model, we were very nicely to dissect that and saying, yes, irisin is hugely important for that. We don't have a single cell data set from the irisin mice at that time when we did the study. So yeah, but I mean, it's an interesting question. Maybe it's something we should do at some point.
Great.
I just had a follow-up question related to the last slide that you presented. I'm looking at my notes here, the SCD2, right? Can you just go back to that slide for a second? Because, that enzyme is essential for producing key monounsaturated fatty acids, right? Like 24-1. And I recall an earlier slide you presented a rodent model using diffusion tensor imaging, fractional anistropy.
Yeah.
And seeing some effects there. So in nirvanic acid, but my understanding is the most abundant monounsaturated fatty acid in myelin, the phospholipid bilayer.
Yeah.
So I'm wondering, in those two studies, and they may be the same. I'm sorry, I didn't catch the
No,
reference.
they're different studies. These were
Yeah,
different
yeah.
studies, but yeah.
Whether the effect of exercise is working through this pathway and leading to more abundance of nirvanic acid, and maybe that could explain your fractional anisotropy findings in the earlier.
Yeah, it's very possible. So what we did in those studies, we looked in different brain regions. So we looked specifically in the dentate gyrus, where
Okay.
we saw more in there, where there is also myelination there, but not as much. But when you look in the commissura, where there's a lot of myelinated tracts, in the same study, you see a huge expansion of myelination there. So
Okay.
yes, both probably de novo oligodendrogenesis, so that there's more oligodendrocytes that become mature oligodendrocytes, which are myelinated,
Right.
is contributing. But there also might be just that the existing oligodendrocytes have more stable myelin sheaths, or they're also like demyelinate or like neurodegenerative diseases that have a demyelination component in there. So yeah, and again, I think with exercise, it's not doing just one thing, right? It's doing many things, and we're trying to understand that. And I think my point is always, I don't think we get a pill that's doing all these things for your brain, but if we can get one molecule that's doing at least one of the very important mechanisms, and is like neuroprotective or staves off cognitive decline, I think we already have a huge win. But yeah, important point. Oligodendrocytes are very important. I think a bit overlooked cell type, but yeah, there's still a lot to look at.
Yeah, interesting. Thank you for the response. So, Martin, I see Martin. Is Martin with us on the call here? Martin, I can pose your question here. He's saying that epigenetics, unless you'd like to speak, Martin, epigenetics is a key player.
Yes,
okay.
Martin, yeah.
My question is there, yes.
Okay.
Thank you, Christian.
Yeah, so epigenetics, yes, they are important. So we didn't do, as maybe some now you can do quite easily, where you do simultaneous transcriptomic and epigenomic studies. So we didn't do this in our study. And I have to be honest, I don't think we actually looked at HDAC specifically. So this is actually something that, Martin, good idea, we should actually look at. There might be changes in there, might be not. It was one of the many, we have looked at many directions. This is not one of them that we had specifically investigated.
Yeah,
So
your study
thank you for the
provided a
suggestion.
great example for for you identify different genes and different cell types, but HDAC 6 and H11 provide a good molecule that we can inhibit.
Yeah.
If you find any promising data, we can have small molecules for them.
Wonderful, great idea. I like it. Thank you.
Thank you.
Very nice. And maybe one last question. Do you want to close the session with the final question?
Sure, maybe right question, because this is question to everybody who's working that field. Because human brains, adult neurogenesis is very limited, very low amount compared to mouse models. And there's an idea that in the humans, instead of enhancing neurogenesis, but more like injecting or transplantation of neural precursor cells or stem cells are still ongoing. And in that directions, I want to hear your opinion on that. What do you think?
Okay, I do think that based on the data that I know, also more data that's coming out in humans, I think that there is still enough evidence to show that there are neural precursor stem cells in humans, in adult, in aging, and Alzheimer's disease. There's also data there that I had mentioned before that these stem cells get remodeled in a negative way in Alzheimer's disease. The whole niche is, I think Sehun had a really nice review on that, like the whole niche where the stem cells are is affected in Alzheimer's disease. I don't want to say that Alzheimer's is now a neurogenic deficiency disease, but I do think it's, from a plasticity point of view, it's a very strong or very plastic part of the brain, what you get with neurogenesis. And that's why I think it's still interesting. If treatments that increase neurogenesis are going to be good for improving cognitive decline, I think the studies are ongoing. But I think you have to always keep in mind both things. It's not just enough to put the cells there. They also, as Rudy pointed out, they have to survive. They have to be integrated in the network. So you have to make sure you have both. It's the same. Otherwise, what happened in my vegetable garden? Like I put seeds in there and then nothing else, and then in fall, there nothing grew. So you also need you need to have a healthy stem cell niche. And I think again that's why exercise is important, because it not just changes the proliferation of the precursor cells, but it also changes the neuroinflammatory environment. It changes the astrocytes. It changes the blood vessels, which then enhances neurogen.
BDNF is miracle grow for the brain.
I guess that still holds true. And BDNF was also changed in our data set. No one actually has asked that, but BDNF was one of the genes that was changed in mature granular cells with exercise.
Great.
Okay, well, thank you for that. And thank you for the questions and the discussion. Dr. Wrann, thank you so much for coming back and presenting updates. Congratulations on the new papers and the new data. And we hope to have you back sometime in the future.
Thank you very much.
Okay.
Thank you, Christiane. Excellent.
Thanks a lot, everybody. Have a great week.